Science Seed, a partner amongst the multidisciplinary consortium of Microphore Biogas, who are on a mission to optimize biogas production by optimizing the microorganisms responsible for it. In previous episodes of this podcast, we have explored the world of biogas production from different angles, the European economic transition towards a greener future, the history of this interesting flammable gas, the process of anaerobic digestion, and most importantly, the role of the microorganisms involved in it. But the truth is, we don't know very much about them. Historically, the processes happening in biodigesters have been like black boxes, with little known about what occurs inside. And as we all know, information is power, almost literally in this case. So if we tame the microbes involved in biogas production, if we conquer the mechanisms happening in their environment, you will have an optimized, reproducible way to produce biogas to power our lives. And that's what we're trying to do at Microphore Biogas. In our very last episode, we explored surprising connections between probiotics, poop transplants, and biogas production. All these processes involve bioaugmentation, which we're going to be talking about today as well, which is boosting the relative quantities of beneficial microorganisms to improve the outcomes. And it's all thanks to these tiny microorganisms playing crucial roles in each process. What's more, in this online event, we are bearing great news because our researchers have discovered a new order of bacteria that might just be a game changer in the production of biogas. And we're going to tell you all about it today. So I'd like to introduce you to our three panelists today. We have three amazing guests who have made wonderful careers out of the study of microorganisms. First, we have Adriel Latorre, which is a doctor in biotechnology, director of the Genomics Department at Darwin Bioprospecting, and also one of our partners in the Microphore Biogas Consortium. Welcome, Adriel.
Hi, thank you for having me.
Thank you for being here. We're also joined by Manel Porcar, who is also a doctor in agronomic engineering, a senior researcher at the University of Valencia, and the CEO of Darwin Bioprospecting, and the scientific director of Microphore Biogas. Welcome, Manel. Okay, and finally, also representing Microphore Biogas Consortium, we have Pascal Otto, a doctoral researcher at the Institute of Waste Management and Circular Economy at Dresden University of Technology. Welcome, Pascal. Sorry.
Hi, thank you for having me.
Okay, so we're going to start the discussion by understanding microorganisms. Back to the basics. I'm going to ask you some questions about microbiology. Before we start getting into details, we're going to get back to the basics. In the frame of biogas, you've probably heard about methanogenic archaea, anaerobic bacteria, and basically the different types of microbes inhabiting biodigesters. So, Manel, could you explain to us the difference between these microorganisms, bacteria and archaea?
Yes, sure. So in any biogas reactor, of course, you have tons of microorganisms. You have a lot of microorganisms, and you have from viruses to fungi. But the main key players that are working for us to produce biogas are bacteria and archaea. So I think that most people do not know that they are dramatically different microorganisms. So first, maybe we could start with the similarities. So bacteria and archaea are microorganisms. So they are living entities that cannot be seen by the raw eye. You need a microscope to see them. But even if they have similar size, sometimes similar shapes, although some archaea have very odd shapes, they are very different because phylogenetically, this means that the origin considering the time from which they have been differing for a very, very long time. In fact, metabolically speaking, archaea are very diverse, and in some metabolic aspects, they are closer to us, to eukaryotes, than to bacteria. So we can then summarize maybe by saying that archaea and bacteria are microorganisms in both cases, but they are very different. So they are like cousins, but they are not like brother and sister. And of course, in the biogas facilities, you can find both because they are both required to produce biogas. So biogas is a mixture of methane and CO2, which is ultimately produced only by archaea. But in order for archaea to be able to produce this methane, this biogas, they need to be in good company, and that company is bacteria. So to make a long story short, what happens is that the microorganisms, particularly bacteria at the beginning, break down the organic components of the biogas facilities. It can be sludge, it can be wastewater, it can be crop residues, whatever. And from those molecules, acid is produced, acetate is produced, and finally, archaea and not bacteria convert these products into biogas. So I think that's the main difference between, there's quite a lot of difference. And the only thing they have in common is that in both cases, they are tiny and you need a microscope to see them.
They're unicellular and
Yes,
like
of course,
just
they are
tiny
unicellular.
creatures.
It is true that there are many other microorganisms that are not bacteria nor archaea that are unicellular as well, and they are eukaryotes. But of course, the thing that they have in common, archaea and bacteria, is that they are not eukaryotes. They do not have a nucleus, and they have all the genetic material, let's say, floating inside the cytoplasm. But this is just one of the similarities. When you compare the genomic identities, the similarities between one bacterium, one random bacterium, and one archaea, they are super close, super far away one from the other. And in fact, some archaea are closer to eukaryotes, to us, than to other bacteria. So it is not because they are tiny and they are microscopic that they are close one to each other. And as I mentioned, they are very different and archaea are closer to us, to eukaryotes. And from an evolutionary point of view, in fact, the eukaryotic cell is a mixture of both. It's, let's say, some people say that it's a bad digestion that one archaea had that included some bacteria it was eating. And finally, this combination of bacteria and archaea ended up being an eukaryotic cell. And then by combining several eukaryotic cells, you ended up by having a plant, a dog, or a human being.
Interesting. Thank you so much. So Pascal, I think Manel has already introduced this, but I wanted to ask you if you could explain to us a bit more what is their role. So we know they're all present in the biogas making process, both archaea and bacteria, but what are their specific roles? And maybe here we can talk about more, introduce our audience to the concept of anaerobic digestion a bit deeper. So what exactly do archaea and bacteria, what do they do inside these biodigesters?
So they have like different roles in this whole process. And this process starts like with big organic molecules. So we have like carbohydrates, we have proteins, we have fats that go in this process, and these are degraded. And the degradation happens in 4 steps. And in the first 3 steps, there are bacteria involved. In the last step, the archaeas are involved. And yes, in the first step, these big molecules are hydrolyzed. So this stage is called hydrolysis. And after this step, these like there's the acidogenesis where the acids are produced. After this, the bacteria use these acids to produce acetate. And the last step is the methanogenesis. In this step, the archaea are involved. And they use either acetate or carbon dioxide and hydrogen to produce the methane that we want in the process. So they have really different roles. I think archaea are the ones that are mainly involved in the methane production, but you need the bacteria before to clear the path to produce the methane.
So it's kind of like a symbiotic relationship. We're going to be talking more about this a bit later, but yeah, I think it's really interesting to, have the differences clear and know what each of them do in the biogas making process, and how do they interact with each other, because they need each other. You need a lot of microorganisms together doing their job and working together towards making biogas. And this is the beauty of it, and what we're doing in Microphore Biogas, this bioaugmentation, is to define which ones can help us better to produce biogas and like feed the biodigesters with the consortium of microorganisms that can optimize the production of biogas, right? So it's really important to understand their exact roles and how they interact with each other. So now that we understand the big differences, I wanted to ask Adriel, why are these microorganisms so important to our lives? I mean, knowing that you've studied all sorts of microorganisms, you're a genomics expert. I think you have experience in other microorganisms other than the ones that produce biogas. So if you can think of other uses or other benefits that microorganisms have in our lives. If you could
Yes,
tell
of
us more about them.
course. Of course. I mean, maybe microorganisms are everywhere. I mean, they are literally everywhere. Even in the most harsh places in the world, they live there. And maybe the audience is more familiar to pathogens, which are the billions of the movie, this microbial movie. But there are many microorganisms that have been used for centuries, even for thousands of years, for biotechnological applications. I mean, for example, microorganisms are used for preparing foods like bread with this Saccharomyces cerevisiae, or wine, or even beer, kombucha. There are many foods that are based on microbial transformations. Beyond that, then we have, and we also studied this in Darwin, we have the human gut microbiome, that maybe the audience is familiar to these two. I mean, the microbiome, it's the human microbiome, they are the microorganisms that live inside our body, on our skin, in our oral cavity, and the most famous microbiome, I would say, it's the gut microbiome. So there are these microorganisms, bacteria, some archaea too, fungi, viruses that are living inside us, and that they have a very important role in our health. For example, they produce vitamins. They help us to with the digestion of fibers, or there's even it's a very hot topic at this moment, what it's called the gut-brain axis. So even the bacteria that are living in our gut, they are producing some compounds that activate some routes that reach our brain, and they are able to have a response in the brain. So there are bacteria that have been associated with a lower prevalence of, for example, anxiety, let's say. So as you may see, bacteria can be used for, if we have like the biotechnological vision, we can use bacteria, we can use archaea, microorganisms for almost every biotechnological application that we may think of. For example, scientists, and even we in Darwin, we are using bacteria for example, for environmental projects like degrading plastics, that they are very difficult to degrade. But there are some bacteria producing some enzymes that can be used or even tuned to be more efficient for the degradation of plastics, or for the degradation of other pollutants like benzenes or other hydrocarbons. So yes, I would say that the microbes are everywhere, and they can be used for many biotechnological applications.
That's amazing. for everyone that is listening to us, I would really encourage you to check out the work that they're doing in Darwin. They have a lot of interesting publications, all to do with microorganisms and the beneficial impacts and things they do in our lives. And it's a very, very interesting world out there in the, micro level. So, okay, now we understand microorganisms better. I want to get into how you go about discovering them, because we are here to talk about a discovery. And I am sure we've all seen news about the discovery of new species, scientists describing a never-before-seen insects, new marine species. And in your case, Manel, you have a lot of experience with new microorganisms, hunting them from the comfort of a house kitchen to the depths of the Mediterranean Sea. So can you tell us how one goes about discovering new microorganisms? What are the first steps or things to consider?
Well, I think the first thing that we must consider is what Adriel was saying, that microorganisms are everywhere. So the number of microbial species in our planet is, as we know, is huge. It's almost infinite. It's like 10 to the 12. It's an expected number of species. But this is just refers to the species. So if you consider the strains, subspecies, etc., the number is almost infinite. So this is good and bad at the same time, because biotechnology is using living beings for a useful purpose. So it can be farming, plants, crops, cows or rabbits, whatever. This is biotechnology as well. And of course, genetic engineering and synthetic biology, which is basically what people think of when you say the word biotechnology. But there's also something in between the traditional farming and synthetic biology, the cutting edge, let's say, biotechnology, which is biodiscovery. So looking in the nature and looking for microorganisms that are already there, that have been somehow shaped by the power of natural selection, in order to select, once again, in the lab, the ones that can be useful for making many things. So Adriel was just highlighting some cases like bioremediation, which is the removal of plastics by selecting those microorganisms that are actually able to eat plastic. But then you can identify from very specific surfaces like the solar panels on from desert microorganisms that can produce carotenoids, and those carotenoids have applications in biomedicine. Of course, probiotics. So most people that go to the supermarket and buy yogurt, like we're not going to talk about brands here, but very well-known shots similar to yogurt, do not know where those microorganisms come from. And those microorganisms very often come from baby feces, baby shit. So this is one possibility, to study the human gut, animal gut, fermented vegetables, so the list is very long, in order to isolate microorganisms that can be used, for example, as probiotics. But when you say probiotics, it's not only for human, it can be for animal as well, because animals, as we are animals, so they may need probiotics to fight against, iron deficiency or some nutrient deficiency, fungal pathogens, whatever. So the same kind of diseases that we human has, even mental diseases that are known to affect our friends, the pets, cats and dogs, for example. And finally, you can consider something which is similar to probiotics, but is applied to plants. So the equivalent of probiotics for plants is what is called PGPR or PGPY, which stands for plant growth promoting rhizobacteria or plant growth promoting yeast. So those are bacteria and or yeast that when you apply them in a field, they get in close contact with the roots of the plants, and they can dramatically foster the growth of the crops. And we know this very well because in Darwin, we have selected a bunch of them, and the results are simply spectacular. So you can imagine, of course, this is a podcast, we cannot show here the plants, but you can imagine one lettuce, which is the control one without those microorganisms, and then you have an average lettuce. And then when you supply some specific microorganisms, then your lettuce is almost twice as big. So it's amazing. And well, there are a lot of reasons for that, because they are fighting fungal diseases, because they are helping the plant to get more nutrients from the soil, because they are fixing environmental and nitrogen from the air. They are solubilizing phosphate, etc. So the world of microorganisms is huge. And just to answer your question, Adriana, how do we go out there to look for them? So it depends on what we are looking for. So if we are looking for microorganisms that are able to degrade plastic, a good idea would be to go to a place which is very rich in plastic and yet is very poor in other compounds, in such a way that the microorganisms, they are very likely what they are eating is plastic, because they do not have much else things to do to eat. If you are looking from probiotics, you should go to babies that are breastfed, for example. This is a good choice. And then you take from the feces some samples that you can isolate. And then, of course, you have to verify them that work. And just to give a final example, if you want to find microorganisms that are good for fostering the plant growth, this PGPR or PGPY, well, there are a lot of strategies, but one possibility would be to check some specific varieties that are able to survive in very harsh conditions, or to select some specific plants that are incredibly healthy and big, and then try to study which microorganisms are associated with those plants, and maybe, hypothetically, of course you have to confirm that, responsible for this increased health and yield. So that will be just 3 examples of which kind of strategy one can follow to try to enrich your collection in microorganisms with industrial interest or potential.
Amazing. So you gave me the perfect introduction for my next question, because indeed, what do you do when you're looking for microbes? You have to choose a place to look for them. So here in Microphore Biogas, what you're trying to do is to optimize the microbial communities that produce this gas, this renewable energy. So where do we look for the microbes? Of course, in biodigesters. And what you did was to get samples from 80 plants around Europe, if I'm not mistaken, and you sequenced all of the microbes in them. And well, I'll let Adriel explain this better. So this is how you discovered this new order of bacteria called Darwini bacterialis. How did you find this new bacterial order, Adriel? And if you could tell me as well what the implication is to the project.
Yeah, you were right. I mean, in your introduction, you mentioned that biogas production is it's considered a black box process. I mean, there are many and many scientists in the world studying these processes, and I mean, it's slowly starting to be less a black box process. But there are many questions to be answered, because biogas production is also a very complex process in which there are many parameters that are involved. I mean, they are using different substrates to produce the biogas. They're using manure, industrial waste, urban wastes, plant wastes. So there are very different substrates, very different type of reactors. Parameters such as temperature or pH changes a lot. So all of these parameters affects to the microbial communities. So one of the objectives of Microphore Biogas were to study how the industrial reactors that are, let's say, hard to study because they belong to private people that use the reactors to produce energy. So they are not so well studied. So Pascal, the University of Dresden, and in help with in collaboration with BioClear, that it's also another partner of the project, they sample 80 different microbial biogas reactors across Europe, mainly in the Netherlands and in Germany. And then we apply anomic technique that is called metataxonomy, and another that is called metagenomic, that they are very related, and they are used to study the composition, the microbial composition of a sample. Let's say with metataxonomy, we can obtain a list of the microorganisms that are present in a sample, and with the metagenomics, we can also study their genomes. So In combining these two techniques, we were able to find out that the most abundant and prevalent bacteria in these reactors, in these industrial reactors, was a bacteria called MBA03, that now we have proposed to name this bacteria Darwinibacterialis, so it's the same bacteria. The first thing that came into our minds was like, how in the earth a bacteria is called MBA03? I mean, it's a very strange number for a bacteria. So it meant that this bacteria was not so well studied, because this name, the MBA03, was a provisional name. So for us, was surprising to see that a very important technology, that is biogas production, the most abundant bacteria and the most prevalent, because it was present in the 80 samples that we studied, was not so well studied bacteria. So then when we applied metagenomics, we thought we have to study in depth this bacteria. What was the problem? The problem was that there was not a single genome in the public databases related to MB03. So in practice, it was not, we couldn't detect this bacteria, which was very strange, because I mean, we have this metataxonomic signal that says that it's the most abundant, but then when you do the bioinformatic analysis of the metagenomic data sets, you cannot find the MB03 genome. So it didn't make sense. The thing was, what we thought at the moment, and we were right, is that this bacteria hasn't have a genome sequence. So the genome is not in the databases. So if the genome is not in the databases, we cannot find this bacteria in our data sets. So what we did at the moment was to go fishing the genome from the data set, and we were able to recover, I don't know the exact number, but I think it's more like that 100 of different genomes of MBA03, and then we were able to study this taxonomic this taxon, and we found out that it was a whole new order of bacteria. I mean, for the audience, the bacteria are organized in taxonomy that starts from the domain, karyotic or prokaryotic, then you have the phylum, then you have the class, and then you have the order, then family, genera, genus, and species. So it's like a whole new order of bacteria that went unnoticed until we did this experiment. And the main reason of this lack of studies on this bacteria that now we propose to call Darwinibacteriales, it's because it cannot be cultured in the laboratory. So people that was culturing bacteria and microorganisms and archaea from biogas samples in the past, they couldn't culture this bacteria. So at this moment, it only can be studied from an omic perspective. So that's the reason we were the first one. I mean, we weren't the first one to know that this bacteria exists, because someone in the past named it MBA03, but we were the first ones to be able to recover the genomes and study this bacteria, bacterial origin in depth.
Nice. And I wonder, this question is going to be for Pascal, because Adriel said this bacteria hadn't been properly sequenced and characterized before. We knew it existed when we found it, but we were the ones who characterized it properly, and now we're studying its role in producing biogas. So can we assume it's essential for biogas production if it wasn't studied before and we found it in all of the 80 samples from biodigesters? Pascal.
So, for sure. Yes. So the reason that we found it in all the digesters gave us the hint that it needs to be essential for the process. So it's a problem that we can culture it, but it is so abundant in also a lot of high-performing digesters that we sampled it must be the correlation that this species or the Darwini bacterialis is related to the performance of an anaerobic digestion plant. And what we found out also digging deeper in the topic of this new order is that it could be related to the heterogenotrophic metagenesis, which is the methane production with CO2 and hydrogen. And this MBA03 or Darwini bacterialis is related to that and acts as a syntrophic acetate oxidizing bacteria. So it provides the hydrogen and the carbon dioxide for the methanogens and acts as like a syntrophic partner to the production of biogas. So it forms this syntrophic relation. We don't know about if there are any more, but we can assume that there are more syntrophic relations that they form. And yeah, maybe for the audience to explain what syntrophy is, it is a cooperative metabolic interaction between at least two species, and they to try to degrade like a substrate, a specific substrate. And this syntrophy happens a lot in the anaerobic digestion. So all the bacteria and also archaea needs to act with each other and produce the intermediates for the next step. So this is a really important point in anaerobic digestion.
Amazing. Yeah, this is exactly what we were saying before. In every process, you might think in biogas production, the key players are methanogenic archaea, right? Because they are the ones that produce the methanogen that we're interested in biogas. But they don't work alone, and they need these interactions to produce biogas. So it's really interesting how in biology, everything works in a collaborative manner. So yeah, it's all about the symbiosis that you were talking about, Pascal. And I wonder, my next question is to Adriel. If Darwini bacterialis produce the food for the methanogenic archaea, like Pascal explained to us, could we isolate these compounds, these metabolic compounds that they give to the archaea to develop a substrate for feeding this archaea? So could we surpass the role of this bacteria and help the archaea produce the biogas directly?
Yeah, it's a good question. I think not, because of the importance of Darwini bacterialis is the syntrophic relationship that we are talking on. So the food, as you say, it's very simple. It's only CO2 and hydrogen. So there are, I mean, very well known compounds that you can supplement to the digester, but it doesn't work at all. I mean, the key here is this syntrophy between the bacteria, in this case, Darwini bacterialis. There are some other syntrophic acetate oxidizing bacteria, apart from Darwini bacterialis, that we are also studying. But mainly, let's say, let's focus on Darwini bacterialis in this case. The syntrophy is key because you have to think on an anaerobic digestion like a process in equilibrium, that it's always looking for this balance. So it's not a matter of adding the substrate, but it's a matter of keeping the microbiome like balanced. So, for example, in this case, the syntrophic acetate oxidizing bacteria, they are taking acetate, that is also a very simple compound, and they are breaking down the acetate to CO2 and hydrogen, and they are giving, or I mean, let's say, they are throwing these compounds into the digester, and the archaea are taking this to produce the methane. So the balance is always maintained, and we think that's the key that's key for the process to keep this equilibrium, and it's the most difficult actually, because we are focusing right now in this pathway, but there are hundreds of different species in the reactor that are also producing different compounds that can be that can activate other routes. So the balance can be can break at any time. So this is why the anaerobic digestion process, the biogas production, is very difficult. It's a very difficult process, and it's also very difficult to study. But answering your question, I think that we weren't able to surpass the action of Darwini bacteriales, because the first results we have are pointing into the direction that the syntrophy between the bacteria and the archaea is needed, and it's key for obtaining a high performance reactor, actually.
So basically, the interaction is crucial, and the presence of both is crucial. And I understand that if we're aiming to optimize biogas production, we're going to making all this possible by bioaugmentation, right? So my question for Pascal is, we discussed in our last episode of the podcast, for the audience, if you haven't listened to this episode, they're all available on Spotify. So in our last episode, we talked about bioaugmentation and what it consists on. But if Pascal, if you could refresh our memory, and for those people in the audience that haven't maybe listened to that episode and don't know what bioaugmentation is, I think it's a crucial concept in what we're talking about. So if you could explain a little bit more and how this process was involved also in the discovery of Darwini bacteriales and the experiments we're doing now.
Sure. So bioaugmentation is basically the targeted introduction of a specific strain into the microbiome of an anaerobic digestion process. So, and the targeted strains are isolates that you culture in the media and then put into this system with a lot of other microorganisms. And the idea is that this targeted strain can help the process. So, for example, if a process is lacking a microorganism like Darwini bacterialis, you can introduce this strain into the microbiome and help the process become more efficient. And this is mainly also the idea of microbiogas. And this was the reason also for the sampling. We sampled a lot of these digesters, 80 in total, a lot of different digesters also. And we also used the samples to create isolates. And these isolates contains a single strain from this process that contributed to the anaerobic digestion. And in total, we selected, I guess, 100 isolates. And these isolates were then tested in bioaugmentation experiments. So we cultured these isolates, and then we put them in a process of anaerobic digestion and looked into the microbiome if this strain is adapting to the circumstances and conquering the process.
Nice. Well, thank you all very much for your expertise in this matter. I want to conclude the conversation with a few more questions. I know the three of you, as you've shared, have experience studying microorganisms from a very different environments. Manel, you've spent a great deal of time studying microorganisms from all sorts of places, from the gut of Spanish people to car tank lids. Pascal has studied many biodigesters around Europe, together with Adriel, who also looks for them in the human gut and even in microwaves. So my question, I guess, for you, Manel, you've already told us how you go about deciding where to look for these microorganisms and why the varied locations, the reasons behind looking for, like, choosing these locations. I wanted to ask you about, because in the world of discoveries, we know a lot of microorganisms have been discovered by serendipity, and some of them were targeted discoveries. So what's your experience with this? To say sometimes, is it more common to find challenges in finding what you're looking for and maybe like finding something else completely unexpected?
That's a nice question. I think that's, as a researcher, one of the reasons why we do what we do is because of curiosity, and we want to know what's hidden in specific places. So, for example, when we started working with solar panels, which was something that was practically not studied up to that moment, we made like some competition to see what we were expecting to find there. And there was one specific taxa that we proposed that finally we ended up finding quite a lot of them in that, and it was Deinococcus. So Deinococcus is this microorganism that is known because it resists super high doses of radiation. So you can explode an atomic bomb, and at the end, you're going to recover some Deinococcus that are going to be happily living there. And we had the hypothesis that these microorganisms, since it is super resistant to radiation, would be living maybe on solar panels because it has a lot of oxidative stress, dehydration, very high temperatures, because of course, in summer here in Valencia, if you touch something which is metallic or glass, which is under the sun for 12 hours, it's super hot. I mean, 60 degrees, something like that. And then we found that. So it was like a prediction was accomplished in that case. But in many other cases, what we find is super surprising. So one example of that, one publication that we made like a couple of years ago, is the microbiome of an ice machine that was very, very close to my office. When I say very close, I mean 5 meters from my office. So it was the ice machine from which we took the ice to work in my lab in the University of Valencia. So it was serendipity, but it was spectacular serendipity, because very often there was like a lot of water in the corridor of the institute. So, and there was someone there cleaning the water. And so after several times that happened, I asked the technician in charge, what's going on there? Why is this machine working so wrong? And he said, I don't know. There's like a kind of mucus inside. I said, what? Let's study that. It looks like a biofilm. So we studied that. It was amazing. It was a jellyfish-like biofilm in the pump of that ice machine, and it's an amazing environment. So when you say the word exotic, you think that we're going to go to the jungle. That's great. Sometimes we will not want to do that, but it can be very exotic to be living from a microbial perspective, of course, in an ice machine, in a water which is almost pure, because it's the water that comes from the ice melting. It's at 0 to 4 degrees, and then you have almost nothing to eat. And then those microorganisms were able to have a consortium combined with fungi and bacteria. And together they were forming this massive jellyfish-like biofilm. And we found a lot of new species. So there was full of diversity, full of new fungal and particularly bacterial species just living 5 meters away from my office. So the take-home message is that in terms, when it comes to microbial diversity, to new species, to the unexpected, very often you don't need to fly to travel to exotic destinations, to the jungle, to the deserts, to the North or South poles, but you can find a lot of diversity just in exotic environments from the microbial perspective, which is micro niches in which the temperature, the nutrients, the radiation are extreme. And this can be very, very close to where we usually live.
Amazing. Yeah, well, Darwini bacterialis was kind of serendipitous, right? You were looking for something, but then to find this new bacterial order that could be so crucial in what we are studying and what we're trying to optimize, which is biogas production. It's amazing. Surprising.
It wasn't expected.
So in your experience, Pascal, what are the key challenges faced when searching for new microorganisms and defining their roles and their nature in the environment they live in? And maybe you can talk about, in particular, in the case of Darwini bacterialis.
So the key challenges, I think, like Manel said, you can find everywhere new species. You need to have a little bit of luck and a good eye for that. But to like pick species or microorganisms that have like a meaningful impact, you need to add value to the discovery you made. So you need to ask you questions like, where did we find this type of strain? How abundant was it? And also, did it occur with other strains? And all these questions arise when we had like this big list of microorganisms that were present in the 80 digesters. And you need to find some correlations between them. So I think the challenge is to find these correlations and see if a certain strain have an impact on the process, and also have a look on like the circumstances and the environment these strains are in. So I think this was like the most challenging thing about the new discovery.
And in particular, I think you mentioned earlier, or maybe it was Adriel, that these bacteria, in particular, Darwin bacterialis, are very hard to cultivate in the lab because of the conditions they live in. Is that something, you know, in the frame of biogas, and you work a lot in waste management as well? They're very harsh environments. Is it really usually you find this challenge where you can't cultivate and study in the lab these microorganisms that you're working with?
So especially species that have like a lot of relations, syntrophic ones, are really difficult to culture. And I think you find every rare species that you can find with sequencing, but you won't be able to culture them. So this is like will always happen with that. But what you can do is you can try to enrich so if you know how the strain works and what it needs, you can try to enrich a sample with this strain if you adapt the conditions and favor the conditions for the growth of this certain strain.
This links very well with my next question, actually, because I wanted to ask about the next steps that were taken or that need to be taken after making a discovery such as Darwin bacterialis. So, Adriel, what are the questions that you start asking yourselves as microbiologists after this discovery, and what are the experiments that are taking place right now at this final stage?
Yeah, the link is perfect because I mean, one of the big I wouldn't say problems, I will say challenge of Darwin bacterialis is that it cannot be cultured yet. I mean, we are trying this, but of course, to have this, it's not a strange thing. I mean, like Pascal said, have said, I mean, almost I don't know, maybe more than 95% of bacteria that can be detected by omic techniques cannot be cultured in the laboratory. So this is very common, especially in syntrophic relationships and in anaerobic environments that are difficult to reproduce in the lab. So this is a big challenge because having a bacteria, in this case in pure culture, it helps a lot to characterize this bacteria. We can do lab tests and see how this bacteria is responding to different substrates, what kind of compounds the bacteria are producing. So this is a challenge because right now what we know and what we are hypothesizing is that this bacteria is present in all the reactors that we have studied. So it has to play a very important role in the anaerobic digestion. There are some articles that are pointing into this direction, but we don't have a direct proof, let's say, that this bacteria actually it's involved in a higher biogas production. So it's something that we want to demonstrate as next steps. So Pascal already said the answer. I think that the solution in this case is to enrich Darwin bacteriales. So we know that culturing this bacteria will be very difficult because we've been trying and we are trying. But enriching, it's not so difficult because we have the genomes from these data sets that we collect from microphore biogas, and we have analyzed the genome. So we have analyzed the functions, the pathways that are present in the Darwin bacterialis genomes. So we know which substrates can be taken by Darwin bacterialis. So we have different ways of enriching this taxa. This is what we are doing right now, and we have started an experiment in Aras de los Olmos, which is also a partner of the project, in which we are comparing the efficiency of two different reactors, one enriched in Darwin bacterialis and one that is a control reactor and has not been enriched in Darwin bacterialis. So we are really looking forward to having these results and to analyze the results, because this will be the next step. And for us, I mean, we are enthusiastic about this bacteria and these results, and we are confident that we are going to find really interesting results in this case.
Amazing. So, well, stay tuned for what's coming in Aras de los Olmos. We're entering the last stage, the last year of the project Microphore Biogas. So this experiment that you just told us, Adriel, is part of testing everything that's been discovered and getting results and analyzing the data and, basically putting into practice and optimizing biogas production, which is our final aim. So I wanted to thank all of you for joining us. It's been an amazing discussion. I hope everyone in the audience that joined us have enjoyed it. And yeah, as I said, stay tuned. In our next episode, we're actually going to see how things unfold in Aras de los Olmos. So keep an eye out for this. And we're going to talk more about new discoveries in the world of microorganisms. Because as we've seen, these tiny creatures have the power to change the world. So thank you all. And it's been very interesting. And we'll see you soon.
Yes, indeed.